Have you always had a burning desire to know more about protein synthesis? Ha ha! Well, the first step, of course, is mastering the vocabulary. It's me, Moser, and after this video, you should be able to correctly describe protein synthesis as the process that cells use to build proteins. You should be able to identify key terms related to protein synthesis, correctly label structures and chemicals related to protein synthesis. It'd be really great if you already knew what DNA was and, how base pairs match up, maybe a little bit about mutations, stuff like that. So what is protein synthesis? Well, it's the process that's used by all living things to make proteins. When I say all, I mean blue whales to bacteria, earthworms to maple trees, ferns to crayfish, and even humans. All these things make proteins in their cells. That's protein synthesis. Let's break down the two words that we're talking about here. We'll actually start with the second word, synthesis, but we're going to talk about what it means to synthesize something. To synthesize something is, it's to make something, to build something, to put something together. The dictionary says to combine or produce by synthesis, to produce something. And in this case, we're not making DNA, we're making proteins, but we're making them from the instructions in DNA. Now, do you think you know what a protein is? Let's check in on that. When I say protein, you might think about meat and beans and eggs and cheese, but in reality, protein's a lot more than that. So it's true that protein does form muscles. Well, meat is muscles. And feathers in bird species are largely made of protein. But proteins also do things like help your body to clot, act as triggers for processes that happen within the body. Proteins also provide important structural components of some kinds of cells. Proteins act as gateways in cells. Here we've got some proteins that are buried in the cell membrane that actually act as little doors to let things in and out. And proteins act as something called enzymes, stuff that makes other processes happen. We'll talk more about those later, but proteins do all these things, so it's not just meat and cheese and eggs. And again, for all those kinds of proteins, from muscles and feathers to enzymes and cell gateways, living things use this process, protein synthesis, to make their proteins. So amino acids, have you heard this word? Well, this is a bunch of, it looks like brightly colored beach balls linked by little curly black things. Those brightly colored beach balls represent amino acids. And amino acids are small chemical units that can be linked together to form a larger important chemical. I feel like this is going to come back to proteins. When we have a chain of those amino acids and they're joined together with those little, again, black squiggly things, those are actually peptide bonds. Well, what are a bunch of amino acids linked by a bunch of peptide bonds in actuality? They're a protein! Well, they're a protein after they've been folded. And when I say folded, we take some amino acids, we hook them together with some peptide bonds, and then, whoa, holy cow! That's a protein, folks. Proteins are folded in weird, wonderful, and amazing ways. Yikes! They have to be. And if they're not folded just right, they don't do their job right. So let's look at one of your vocabulary words. Transcription. Transcription is the process where DNA code, the information contained in DNA, is copied over into another molecule called mRNA. So DNA gets copied over, the information is transferred to mRNA. Both DNA and mRNA are nucleic acids. That's going to become important. If we break down that word a little bit, to transcribe something is to copy it. As a matter of fact, in the Middle Ages, monks spent their whole lives transcribing books, copying them by hand. We can talk about transcribing audio, like a recording of someone talking, to text. We're just copying information. The language doesn't change. Why is that important? Because going from DNA to mRNA, they are both in the language of nucleic acids. We are transcribing. This is transcription. The language doesn't change. So let's talk about that mRNA. The M here stands for messenger. This is messenger RNA. It is the molecule which will carry the message from DNA out to where proteins get built. It's still a nucleic acid. There's an image of one, but it looks a little different. For one thing, where DNA is a two-sided, a double helix, mRNA is a one-sided molecule. For another thing, and this is really important, you remember those DNA base pairs you're supposed to know, A and T and C and G? Well, in RNA, there is no thymine. It's replaced by another base called uracil. Let's talk about uracil. We'll see it more later, but it's the base that replaces thymine in RNA. There's a piece of DNA ready to get transcribed, and when it does that, every place that we would normally expect to see a T, we instead see uracil. What about the word codon? Well, here's a piece of mRNA, and we can look at it in 3 base chunks. Those 3 base chunks are called codons. And when the mRNA is being read, I put that in air quotes, but you can't see them, to build the protein, those 3 base chunks each contain a code that specifies one particular amino acid. Okay, we keep talking about going to where proteins are made and when proteins are constructed. Well, where does that happen? We know it happens in cells, but it happens at a specific little tiny organelle. It's like a mini organ inside a cell, the ribosome. The ribosome is a small organelle in all cells where proteins are made. Whether we're talking about animal cells, plant cells, or bacterial cells. See those little black specks? They kind of look like somebody sprinkled pepper all over my nice, lovely cells. Those are ribosomes, and they're scattered throughout the cell, well, kind of like pepper. It's the place where proteins get built. Okay, we talked about transcription. Now let's talk about translation. You know what translation means with languages. We change something into another language. Well, translation is the process where mRNA code gets made into a protein. We go from the language of nucleic acids to the language of proteins. The DNA gets transcribed to RNA, and then that nucleic acid language is translated into a protein language. This is just like translating an expression or a song or anything else into another language. We go from the language here of nucleic acids to the language of proteins. That's translation. Okay, last word, point mutation. This is actually review from our last matrix, but remember that a point mutation is a mutation in DNA that changes a single base, just one. And in the last matrix, we looked at the 3 types of point mutations: substitution, insertion, and deletion. We're not going to deal with those in level 1, but when we start using what we know about protein synthesis to make proteins, we'll see how these 3 kinds of mutations can actually change a protein or not. Okay, can you describe protein synthesis as the process that cells use to build protein? Can you identify key terms and correctly label structures and chemicals related to the process? If you have questions, you know what to do.